In this video, we will discuss about the relationship between cancer-associated fibroblast and epigenetic dysregulation in tumor cells. So, in this case, the graphical abstract are presented here. So, what happens in the regular cell is that regular cell has these proteins, that is ACTD2 proteins are secreted in the regular pancreatic cells, and because of that, this ACTD2 has this tumor suppression properties. So pancreatic cells will be able to behave normally in the ACD2 proficient cells. However, when the mutation occurs, then most commonly mutation in the pancreatic cancer cells are KRAS G12D mutations. When these mutations occurs, then it will cause epigenetic changes in a way that the pancreatic cancer cells will lack these ACTD2 formations, and once it lacks the ACTD2 formations, then it will secrete some cytokines such as BMP2, and these BMP2 cytokines will then affect in the tumor microenvironment in a way that the cancer associated fibroblasts that are formed in the tumor microenvironment will take up this BMP2 and will form this lipid laden cancer associated fibroblast, or cancer associated fibroblast which is rich in lipid, and this lipid can be utilized by the these lipids which are secreted through this ABCA8A will be utilized by the tumor cells for their energy utilizations. It is shown in this figure that there is epigenetic changes that occurs in the tumor cells, that some of those changes related to this metabolic genes and this BMP2 secretions that will cause the cells in the tumor microenvironment to form more lipid-rich fibroblast, or the fibroblast that are formed in the tumor microenvironment will have more lipid in it, and these lipids can be used by the mitochondria of the tumor cells for its energy utilizations. So this is what the relationship that has been established by this paper, that we will discuss in the subsequent videos, that how the tumor microenvironment will get affected by the epigenetic dysregulations in the tumor cells, and the change in the tumor microenvironment that this paper targets is on the cancer associated fibroblast, how the behavior of this cancer associated fibroblast changes, and this upregulations towards this lipid laden CAF is usually occurs through the acetylations of the positions that we have discussed in one of our previous papers, that is this HEK27ME3. So acetylation of these sites will cause more uptake of BMP2, more uptake of this BMP2, and helps them to acquire more of this lipid-rich cancer-associated fibroblast cells, or the term that they have called them as lipid-laden CAF. And here it is, that is written acetylation of this H3K27A3. This will leads to those change in the cancer-associated fibroblasts in the tumor microenvironment, and these things can be inhibited, or this link can be dysregulated using this G-boxin that is used to target OX4s in the tumor microenvironments, or this TCA cycles, which is the energy cycles of the cells, and that also we will discuss in our subsequent videos. And so H3K27A3 acetylations is shown here in the tumor cells, though this kind of modifications that occurs that leads to the epigenetic changes, and that is one of the epigenetic changes are this metabolic remodeling in a way that energy will be utilized towards this growth and proliferation of the tumor cells, and simultaneously the upregulations of the cytokines such as BMP2 that will help to fulfill the requirement of the energy that is necessary for this uncontrolled uncontrolled growth and proliferation of the tumor cells. So I hope you understood this cancer-associated fibroblast and the epigenetic dysregulations in the tumor cells relationship in brief. We will discuss this thing in the upcoming videos. I might have repeated same things 2 or 3 times so that people will understood or the abstract of this paper, and they will appreciate the experiments that the researchers has done in order to find out the relationship between CAF and epigenetic dysregulations in tumor cells. That's all for this video. Thank you for your time.